Hey guys, Michelle Glass here, and we are continuing our special senses discussion, and the topic for this video presentation is balance. So this is one of our 5 senses. I like to talk about it as equilibrium balance. That's kind of the way I was taught this, which your equilibrium and balance is talking about your sensation of rotation. Really, it's your head's position in space, and you're detecting, rotation of the head. You're detecting gravity's pull, so you can tell if your head's forward or backward. And you're detecting linear acceleration, so you know when you're moving or when you're not moving, right? Okay, so that's what we're looking at here. These sensations are detected by structures in the inner ear, so we've seen this before. Remember, we're looking here at the vestibule and the semicircular canals for the sensations associated with equilibrium and balance. The cochlea we've already talked about as part of our hearing video. Remember too that your inner ear has that bony labyrinth on the outside. It has a membranous labyrinth on the inside. In between you have fluid called perilymph, and then inside that membrane you have a different fluid called endolymph. As we are talking about the detection of equilibrium and balance, we're going to see this movement of fluid that's going to stimulate our mechanoreceptors. And we have the same type of mechanoreceptors here. They're still called hair cells. They look very much the same. But here we're going to see that it is that endolymph that's going to cause those mechanoreceptors to move. Whereas if you remember in hearing in the cochlea, it was the movement of the perilymph that caused those basilar membrane to move and bumping those hair cells up against the tectoral membrane. Okay, so we're going to be focusing in first on the vestibule. This is my wonderful rendition of the inner ear. You probably want to make sure that you're recalling in your mind the image from your models in the lab, and also the diagrams that are in your textbook, because they are much better. Your cochlea, remember, is your little snail shell. That is for hearing. And then we have our vestibule and then our semicircular canals. And we're going to be talking about the vestibule to begin. And this is where you're really getting that sense of your head's position, acceleration, pull of gravity is really here, whereas the semicircular canals is more dealing with that rotation. So think of the vestibule as the forward and backward movement of the head, so movement in kind of that linear direction. Now I've drawn here, this would be like your bony labyrinth, right? So if we kind of try to draw inside our membranous labyrinth, in the vestibule, that membranous labyrinth is divided up into 2 like pockets. So you have the utricle and the saccule. So make sure that you're looking at those diagrams. I've just kind of drawn it as one big continuous pocket, and they're kind of separate but linked pockets in real life. Now inside your utricle and your saccule, you're going to have a structure called a macula. The A ending is singular, so maculae with an AE ending is the plural form, so you do have more than one of these. And this is where those mechanoreceptors that are detecting that linear acceleration and gravity are located. Your hair cell is essentially the same as what you saw with hearing, except instead of just having stereocilia, we have this one super tall kinocilium. And so this is giving us kind of directionality. So if our movement, if our bending is in the direction of the kinocilium, then our hair cell is stimulated. If instead the movement is sort of away from the kinocilium, that hair cell is inhibited. So this allows you to know if you're moving forward instead of backward because of this directionality of movement. You don't just know my head is moving, you know if you're moving your head forward or backward. Okay, here's a picture of your macula. So notice we have our hair cells. They're sitting in an epithelial tissue for support. They're synapsing onto sensory neurons. And those cilia, both the stereocilia and the kinocilia, are extending up into a gelatinous layer. So this is kind of a thicker gel-like material, and then sitting on top of that are these like crystalline stones, which are called otoliths. All right, so what this allows you to do is if your head moves forward, those stones are going to move based on the pull of gravity, and because of their thickness, that gelatinous material moves too, and so those hair cells move. Or if you move backward, then those stones are moving backward, and again, gelatinous material is moving backward, and then the hair cells are being stimulated. So I've just put some markers in my bucket to try to give us sort of that visual representation, right? So if I move, let me kind of equally distribute. All right, if I move down, if I move back, and thank goodness they didn't spill everywhere. Okay, got it? Awesome. Okay, oops, too many clicks. All right, let's focus in on those semicircular canals. Now, remember, you have two ears, you have two inner ears, you have two sets of semicircular canals. So the information is being detected on both sides. And you should remember from your lab studies that those semicircular canals are, oriented in different directions. And there is the membranous labyrinth running through those, which is full of that endolymph. And inside that, you have at the base of each of your semicircular canals a set of ampullae. AE ending is, of course, our plural form of the word ampulla. So let's focus in on what that looks like. It's very similar to the maculae. There's just no otoliths. That gelatinous material is pretty tall. You have hair cells that have stereocilia and kinocilium. You have hair cells synapsing onto the sensory neuron. So as you are rotating your head, the fluid inside your semicircular canals is rotating. And as it rotates, it's going to be pushing that gelatinous material. And if that gelatinous material moves, you're going to be bending your stereocilia and kinocilium, and depending on the direction of movement, you're going to stimulate or inhibit the cell. And so then that information from each of your semicircular canals in each one of your ears is going to be processed at the cerebrum. Okay, so that's what I've said here, but this is a little bit more specific. So you have a vestibular nerve coming from the vestibule, taking information from both the semicircular canals and the vestibule that's becoming part of the vestibulocochlear cranial nerve, nerve number 8. So now we know it's called vestibulocochlear because of the two different nerves, because of the structures in the inner ear. That information does travel to the brainstem, and especially when we're talking about equilibrium and balance and rotation, that information makes its way two places: to the cerebellum, which is responsible for position, balance, coordinating movements, and then the cerebral cortex. So that's where we have our conscious awareness. And that's it.